It'll start the video and then it won't start the video. That's crazy. We're going to be doing the follow along notes for transcription and translation. So let's get started. If I can, there we go. Okay, here is the overview for protein synthesis, follow along notes. It's the 3 pages. I've gone ahead and cut them out along the dotted line and taped them together. And what I'm actually going to do is I'm going to go ahead and color in some things while you might be taping yours out and putting it together, or if you're with me, that's cool too. First off, this dotted looking thing right here, this is representing the nuclear membrane, as you can see here. I'm going to color this in mostly for fun. You are welcome to color it in as well. It's going to help visualize, so I suggest you do. That is the point of these notes, is so that you can visualize a process. You don't have to do what I do in terms of coloring. You can do whatever makes your life happy. Also remember that coloring as an adult, which is unfortunately what y'all mostly are, sorry about that at this point, is relaxing. Not probably when you're trying to learn transcription and translation so much, but if you were to get an adult coloring book and do that on your spare time, you know, all that free time you'll have, I'm sure you have a ton of it, then you will find that it's actually a relaxing thing. There's been plenty of studies about it. I'm just going to do a different color on the inside, which looks nearly the same, doesn't it? It's a little darker where I am currently in my house, so I'm going to leave this light on. I don't know if that's a good choice or a bad choice, but that's what I'm doing. So you're welcome to do that with me. So this is the nuclear membrane. Now, what are the processes that take place inside the nuclear membrane? Do you all remember? Remember, we're talking about replication, transcription, and translation in this unit. So the first one, of course, is replication because the DNA is inside the membrane. But this is not about replication. Oops, it's coloring on the paper. This is about transcription. What is the definition of transcription, if you recall from the lecture? Transcription is translating, nope, see, even I messed up. Transcription is transcribing, which is writing down in the same language. We are in the same language. And what is the language we are using? That is the language of nucleic acids. So we have our 4, that's 3. We have our 4 base pairs of adenine or thymine or uracil, depending on if you're DNA or RNA, and guanine and cytosine. So we are in a 4 base language of nucleic acids. Our DNA coding strand is the strand that's going to match the RNA. So here, A, T turns to U, G, G, A, C, G, you know, T, D, U, T, D, U. I'm not lying to you, it matches. And so our template strand here is what the RNA is being built off of. And, you know, just for fun, we're going to go back to coloring. So I'm going to color my DNA strands in green. You can use whatever color you like, so that we can see that the RNA is different. So I'm not going to be super fancy about this because I don't want to waste all of y'all's time. But y'all can color along with me, or you can watch, maybe turn off the sound and just watch, follow with the eye movements. I forget what that's called. That's also supposed to relax you. This was easier when this was a much smaller piece of paper, but since I printed it so big, it's going to take more time. Now, recall, please, that when we, in eukaryotic cells, when we transcribe mRNA, do we automatically get to use that mRNA immediately? You should be answering no, we do not get to use it automatically. And why? And you should be answering about how we have to do modifications. We have to do post-transcriptional modifications to that mRNA. We actually call it pre-mRNA. And then we have to do post-transcriptional modification so that it can be functional or mature mRNA. So our mRNA, when we copy it from our DNA, remember we have a lot of junk in our DNA that doesn't necessarily mean anything to our current lives that we are living on this planet. We have all the DNA in our cells, from all of the evolution of our cells. So we have exons, exons being expressed, remember exons, expressed, and we have introns, and remember introns is in between. We have to get rid of those introns in between. And you know, just to make a point, remember that T is turned to U here. I have a few more things to say over here about the DNA, but since I've already moved on to here, let's do that. So we have to get rid of those introns. So here it is showing you in gray are the introns. And just let me do this. I have an OCD brain. You'll know I'm type A. So let me get some color on these nucleic acids before I move on. We're gonna splice out those introns. Does this light need to be on? Yeah, I think it does. As the first modification. Remember, we do it with a spliceosome with snRNA and proteins, and we loop out the introns and we cut them, just straight up cut them out of the DNA, and we paste the exons together. So here you can see with my colors, we have taken out the introns and now we just have a solid blue mature RNA that is ready to go out into the cytoplasm. Okay, so let's go back just a little bit. DNA, by the way, DNA is a genome. We haven't used that word a whole lot, genome. A genome is your entire library. Everything, every gene, everything that we've ever needed for all of evolution is in our genome. Gene, I mean, think of libraries. How many books are in a library, right? It depends on the size of the library, but hundreds to thousands to millions on some libraries. A gene then, which is what is here in green, in this transcription bubble, is like a book. Okay, and in the book you're reading paragraphs and sentences and whatever, and that's going to be our mRNAs that get turned into thoughts in your head, if we're going to go with that whole entire metaphor. Transcription. What happens in transcription? You already know this. A section of DNA unwinds. And who comes along and binds to that section to do the transcription? Do you remember? You should be saying RNA polymerase, which one? 1, 2, or 3? 2, and transcription factors. I'm going to put and transcription factors and bind, and RNA poly 2 reads, quote unquote, reads the template strand, the template DNA to make a copy. To make a copy. There's 2 words there, to make a copy. So a section of DNA unwinds, and then RNA polymerase 2, along with transcription factors, bind to the RNA polymerase, bind, comma, and the RNA polymerase 2 reads, quote unquote, the template strand to make a copy. We've already talked about the post-transcriptional modification, but there's 3 of them. So we talked about one. The first one is remove remove the introns, right? So if we remove the introns, the exons get expressed. What's the second one? Remember, we have to have something, well, we do two things to make sure that the mRNA, the mature mRNA, is not degraded immediately when it gets out into the cytoplasm. One of those things is that we put that 5' cap on, called the 5' cap because it's on the 5' end of the strand. And what's that last one? We put that 3' polyadenine tail on. And so there's our, here, 3' polyadenine tail, and you can see it anyway. And there's our 3' polyadenine tail. And this just tells enzymes in the cytoplasm to not degrade it immediately, but also as the enzymes start to degrade, you have some time before it gets to the coding mRNA. And once again, all this is in the nucleus. So I'm gonna actually write that. This show up really well. This is nucleus, right? And here we are in the cytoplasm. Good. So that all happens in the nucleus, these two steps. Then we're in the cytoplasm. The cytoplasm, whoops, we are doing translation. This is a different language. Different language. Remember from the lecture that you just watched, this is 4 base pair language. This is a 3 base pair language. We turn into a triplet code, triplet language. Okay. And that's actually very important that you understand that for both your test and your life. Translation, I'm going to say real quick, what we actually are going to say is see other lecture, see other follow along notes. But what you have, you already know, is initiation, elongation. Now what's that last one? How do we stop it? We of course say termination. Okay. So what happens in translation? Well, you have your mature mRNA. And I'm, gonna take my time to do what I need to do. You take your time to do what you need to do. There we go. You have your mature mRNA, and it goes out into the cytoplasm. And remember, it can be sent to the rough ER with a signal peptide, or it can stay free in the cytosol. If it goes to the rough ER, that means that it's being told that it's going to go somewhere else, out inside or outside the cell. If it's free in the cytosol, it's going to immediately be put to work right where it is made. Your rough ER, wow, what? Sorry. What is the very first amino acid we have for every single proton, no matter what? We have 3 letters that code for the start. Do you remember from the lecture? If you don't remember from the lecture, you could be looking at the first 3 letters right here. And then the first amino acid right here. So the first 3 letters for any protein, ever is methionine. That means start. What that means is that this small ribosomal subunit recognizes the AUG that means start and comes and binds to it. Yes, I'm not coloring in lines, deal with it. Once the small ribosomal subunit is bound, it triggers the large ribosomal subunit to come along and bind as well. And when the large ribosomal subunit binds, then translation can begin. But you can't do it with just the small ribosomal subunit. You have to have both. I say ribosomal weird, and I always say it like slightly just a little bit long, wrong, not long. So, I mean, you can call me out on that, that's fine, but I also know that I'm doing it, so don't think I'm not aware of my own fallacies. Oh, the ribosomal subunit's kind of big. Just gonna outline it all just for funsies and also to make y'all miserable. That's really what I live for as a teacher, isn't it? To make y'all miserable. It's actually not. It's just a happy side effect. Okay, so there we go. Ribosomal subunits have come and bound along to that mature mRNA. AUG means methionine. Methionine always means start. No matter what protein you were talking about, it's going to be methionine. And then the anticodons of the other tRNAs code for other proteins. Amino acids, I'm sorry, they code for other amino acids. And so up here you can see tRNAs that have already been through the amino acyl tRNA synthetase. Remember that guy? That enzyme that puts the amino acids on the tRNAs. It's an unstable bond, which is why they're so happy to release the amino acid when they get to the ribosome. Yeah, so they carry, the tRNAs that have been bound to the amino acids, we call that amino acyl tRNA when you have them both together, they come to the ribosome to build the protein. In what order do they come in the ribosome? Do y'all remember? Remember, it's not really showing it here, but there's like 3 little windows in this ribosome. You can kind of draw them in. And they have names. You have the A window for amino acyl transferase. You have the P window, and then you have the E window, E meaning exit. E meaning peptidyl something, something, something. But A is where the new amino acyl transfer tRNA comes in. P is where the growing polypeptide chain is. And then E is where the empty tRNA is exiting from the ribosome. In terms of follow along notes, that's it. We have one more box here. Post-translational modifications. Translational. Now the protein has to fold. It doesn't just stay a chain. It turns into a completely functional 3D, I'll just keep coloring it in orange because the amino acids are orange. 3D shape. Do your worst or best. There we go. Remember, at first it has to, okay, so remember that this is the primary shape, the chain of amino acids. Then it has to form those alpha pleated sheets, or the alpha helices or the beta pleated sheets. Remember those from your first exam? And then it has to bond to amino acids across from it, like disulfide bridges and covalent bonds and hydrogen bonds, and it forms the tertiary structure. And then it can go on to the quaternary structure. Do you remember what that is? Primary to secondary to tertiary and or quaternary. The quaternary structure is when you have multiple polypeptides that have to work together to be functioning, like your hemoglobin in your cells. Some of these are going to go to Golgi to be sent out of the cell. Okay, big long piece of paper for not actually a whole lot of written information. It's more the coloring so you can visually see the steps of how transcription and translation occurs. All right, cool. Thank you so much.